It's my pleasure to introduce Professors Sid Bosch from SUNY Geneseo and Mike Chislock from SUNY Brockport.
All right. Thank you. Well, we just want to welcome everyone to this wonderful poster session. We have a total of 11 posters from students who work at SUNY Brockport and at SUNY Geneseo, either through independent directed studies, or in the case of the Geneseo students, they're all part of a class that runs every fall semester, and we spend a lot of time on Conesus Lake and Hemlock Lake. So we just hope you enjoy the science, and thank you for joining us, and thanks to everybody who worked so hard, not just the students and the science that they did, but also the staff at the Watershed Education Center who have been preparing the technology and everything so we could broadcast the whole event.
Yeah. Sid and I have been doing this since fall of 2017, so we've been doing this for quite a few years now, and we're definitely really happy to be here. It's a good opportunity for the students to get some experience presenting. Most of the students here are in my limnology course. A few students were also summer research students, so we're excited to be here. Thank you for everybody setting up for us, and let's get the event kicked off.
I'm Carl Hannafin, and I'll be helping out with the Zoom presentation. We'll spend about 5 minutes on each poster. There's 11 posters, so it'll be about an hour.
Hi, everyone. So today we're talking about freshwater salinization. Salinization is the process of an accumulation of salts, including chloride, into our freshwater systems. So primarily when we're talking about salinization, we're thinking about terrestrial salt inputs from road de-icing, which is really relevant. We just got our first snow, already seeing salt all over the roads. So from the 1940s all the way until now, we've increased like exponentially how much salt we're putting on our roads every year. Road salts are water soluble, so they'll enter our water, but then chloride will not degrade, which is what kind of the thing that we're most focused on is the persistent contaminant. So some of the things that salinity can do when the salinity of our water is increased is we can have a lack of mixing in our waters. So most of our local lakes are dimictic, but they can transition to being meromictic or monomictic, meaning they're only going to mix in the fall. So essentially, salty water is more dense. It's going to sink to the bottom. And then if that resistance is too big, it's not going to be able to mix in our spring turnover. So this can cause the development of hypoxic or anoxic conditions, which is dissolved oxygen of 2 milligrams per liter or less. And that can influence biotic organisms quite negatively.
Okay, so a little bit about some ecological impacts of salinization or freshwater salinization. So we'll talk about 3 main trophic levels within a lake ecosystem. So a lot of the studies that have been done on zooplankton, which are, I don't know how much people know about different animals within a lake ecosystem, but they're little like animal plankton that swim around and they eat phytoplankton, so algae. So algae has actually been found to be increased in abundance due to salinization. But despite that, salinization does negatively impact zooplankton species, which could have potential impacts on fish, but it has not been found to be significant. So the thing with salinization and fish, though, is that it can be impactful on different life stages of fish. So juvenile fish and fish that are in the egg stage, it has been found that salinity will reduce the female's egg swelling, egg survival, and embryo development, as well as larval growth. So overall, it just kind of messes with the food web dynamic within the lake system. So because of all these issues that we've discussed, we would like to highlight some different management implications for this. So long-term studies have shown that salinization is not going away. Obviously, we continue to get winter storms, we continue to get ice on our roads and have a need to de-ice for public safety reasons. But because of the fact that we aren't stopping salt use, this is just going to continue to be an issue. A lot of studies suggest looking at watersheds as a whole for management of freshwater salinization. That's because you can look at it from a whole comprehensive view of what is going in and out of the system and each ecological effect within the system. It has been found that salt application on roads that are less than 500 meters from the lake has significant impacts on increasing salinity within the lake. Most of the regions currently only protect lakes within a 300-meter buffer. I mean, this isn't everywhere, but that's just kind of a standard for places that do. So there's a clear management need to potentially increase this buffer around lakes in order to improve the amount of runoff from road salt that's getting into them. Another, yeah.
We don't have anywhere near 300.
Anywhere. Yeah. No, I know that's the unfortunate thing is around here, we don't do anything about that.
Can I just ask a question? This red circle here, that represents a high density set of water. Its density increased because of all salt.
Yep.
And that's interfering with the food web? Is that basically what you have?
Yeah, it is interfering with the food web for a couple different reasons. The lake no longer being able to mix because of this dense saline layer. So that kind of prevents the input of nutrients throughout the system. Because mixing does a lot for the system. And then also, because of the amount of salt going into the lake, one of the biggest issues we see is salt has impacts on other chemicals. So other legacy nutrients that might be in the system can then bond with some of the ions that are released when salt goes into water, and then kind of cause these chemical cocktails, as a lot of the papers have referred to them, that are then released into the system and harm the biota in other ways rather than salt directly. So yeah, there's a lot of issues with this dense saline layer down here.
Yeah.
It's not good for the lake at all.
Wish we had more than 5 minutes to ask some questions.
Yeah, I know.
But
I know.
I'm afraid I have to move on.
If you come back around if you get a chance.
Thank you very much.
You're welcome.
Appreciate it. Nicely described. Okay.
I'm Jake Callis from Geneseo. This is Louis D'Ambro, also from Geneseo. This semester, we've been studying the rudd, an invasive species in Conesus Lake, and what they've been feeding on. So we have this freezer full of frozen stomachs that we thaw out and cut open every day in lab, and we sort through them to sort the contents. These are some of the things that we found so far. Some of them identified, some of them not. These are a couple of the unidentified ones. We found a lot of algae, both filamentous and branched, and a lot of invertebrates, specifically zebra mussels and midge larvae. And now here's Louis to talk about this other graph.
So yeah, as Jake said, this is like the main stuff that we found. We could see a lot more plastics that we found, just in the stomachs. And then if we look at this chart, this is like kind of like the breakdown of everything we found. As you could see, a lot of the algae that we found, zebra mussels are growing like in the amount we found in each stomach. I find a lot of them. And I also find a lot of the snails. There's a lot of like the tinier snails. Yeah. And then what we're just looking at is to find if there's any trends. What we want to do in the next poster session is kind of like compare and contrast the different seasons and the different weights of fish. We just got the information from the DEC, so we hope to be able to do that in the next session.
I have a question.
Of course.
What did the room smell like when you were trying to figure out what was in the stomach of these rummies?
It smelled like progress. You get used to it after a little while, but
Because it was fantastic, because this is one of the newer, relatively recent invasive species we have, and we see a lot of. So I'm glad you're studying and helping us understand how to deal or what their impacts will be.
me too.
Nice work.
Yeah.
Good project.
Thanks.
Thank you. You're on.
Okay. Alrighty. So this is my summer research on the impact of microplastic gradients on freshwater amphipods. So I utilized the pond facilities at SUNY Brockport and did a mesocosm ranges of microplastics and then an amphipod versus no amphipod treatment. So I utilized shade cloth in order to regulate temperature. And then we did in situ data collection with the YSI sonde. We also took water quality samples, which I am in the process of analyzing for total nutrients and dissolved nutrients. So we had 30 tanks overall. We set them up so that the microplastics would be treated in concentrations of 0, 10, 100, 1,000, 10,000, and 100,000, which these are the pictures of our concentrations that we made. They're like a bright greenly dyed microsphere. And so we added them to the tanks and we ran the experiment for a little under 40 days. We were measuring to see periphyton growth, zooplankton growth. We also were looking at amphipod populations, and I'm in the process of counting those right now. So overall, the results were a little mixed. There weren't any clear trends. So in terms of the amount of mass that the leaves lost, we added oak leaves to see how oak leaf mass would be declining due to amphipod consumption and whether microplastics would influence that consumption. And overall, there was a general decrease from the amphipod tanks, which are these striped ones and then the solid ones. But there are some fluctuations where leaf mass actually was higher than we would have expected it to be. We also looked at growth rates. And so this is, again, incomplete growth because I haven't gone through all of the tanks. I have them stored in ethanol jars with the periphyton and the amphipods. But it kind of, we were expecting to see like a drastic decrease where the highest microplastic treatment would result in the least amount of amphipods, but that's not really what we saw. And I don't really know what the metric is of that. I think a lot of it might have to do with the fact we made our tanks really productive. We fertilize them every week. And so this is kind of an example. We have a lot of periphyton and it's mostly filamentous algae. And in the future, if we were to rerun an experiment like this, we would definitely add way less fertilizer because we essentially think that the amphipods are going to prefer the periphyton. And so they're eating that. And since there's so much of it, they're not going to be consuming the leaves as much as they would be. So if we were to continue this experiment again, we would do something a little bit different.
I'm so glad to hear that you're studying the impact of these microplastics.
Yeah.
Because it's a real big problem.
Yes, yeah. Yeah, we had a grad student who was working on microplastics. So I got the opportunity to do summer research and we kind of wanted to extend on this. So this was this was my first field experiment. So it was definitely hard and I learned a lot of things that I would do differently.
Last year there was a study somebody did about the shape of microplastics.
Yes.
Were you careful about the shape of those microplastics that you used in your samples?
Yeah, so we used a laboratory bought microplastic. So unfortunately, we had to use microspheres. Environmentally relevant microplastics is primarily fragments and fibers. We haven't figured out a way. We're actually in the process, hopefully, of figuring out a way to make microfiber plastics in the lab so that we don't have to outsource them because microplastics are like really expensive to buy. So we kind of used what was left over from the grad student, but ideally in the future, we would have fibers and plastic fragments of different sizes. We did like density and mass calculations based off of like the range that they gave us. We had like an Excel sheet. I can't remember exactly what the sizes were, but they're very small. Like I feel like the most that we added was like 20 grams to like.
Hi, everyone. So our project over right here is about emerging contaminants in aquatic ecosystems, particularly fluoxetine. It's also commonly known as Prozac. It's an SSRI or a selective serotonin reuptake inhibitor. It's commonly used to treat depression, anxiety, etc. And a lot of these are entering aquatic ecosystems through wastewater effluents that are not able to fully treat the emerging pharmaceuticals. So this has negative impacts on things like photosynthetic activity, their growth rates, population abundance, all sorts of things. So there's really a lot of impacts on what this could do for higher trophic levels, as phytoplankton make up about 45% of global primary production. They serve as food for zooplankton, and then that would get transferred up into higher trophic levels and cause some really nasty problems. So a lot of the studies that we looked at focused on employing mesocosm experiments, where they use different varying concentrations of fluoxetine at the nanogram and the microgram level. They also measured things like behavioral assays, where they looked at the velocity or the duration of movement and the speed of which that they move at. They also measured decomposition across 90 days in this one study at different depths into the in the mesocosms So you can imagine like the epilimnion, the metalimnion, and the hypolimnion is what they would measure the decomposition in. And they did some measurements on cell volume and biomass and overall growth rates. So I'm going to pass it over to Zane. He's going to talk about the results a little bit.
Hi. So, oops, sorry, I don't know if you can see me. So, hi, I'm saying. So, they found some really interesting things. So, overall, they found that there were generally some very negative impacts on the like growth rate and the biomass of two, specifically in this study, two diatom species. So they found that like biomass yield, so in cells, decreased as soon as you hit about 10 micrograms per liter of fluoxetine. And you see the same trends happen with the relative growth rate of each of them decreasing or starting to decrease at 10 micrograms per liter. So they found a variation of these impacts across not just biomass and relative growth rate. They also found these decreases, same thing at 10 micrograms at pigment synthesis. They found some effects on cell density as well as population growth as a whole. So with these metrics that they found with this decrease, they found that they found ultimately that fluoxetine didn't have much an effect on cell volume, which you can see here that the cell volume effect, while it decreases here in this species, here it actually increases as the concentrations go up. So that's because fluoxetine was not found to have any direct effect on cell volume, rather indirect effects based on the other effects that fluoxetine had on biomass and relative growth rate mostly. So
Let me ask you just something about, you talk about 10 micrograms
yes,
as being a kind of a threshold.
Yeah,
that's where
they really saw the bigger,
So-
So if I were to go out into Conesus Lake and measure the water, what would I find, do you think?
I'm not sure about Conesus specifically. I'd have to do tests on the water.
What have you found in similar water?
In similar waterways, so most like eutrophic or mesotrophic water systems have about between 10 to 50 micrograms per liter. So in a lot of systems across the entire world, there are pretty significant levels of fluoxetine nowadays,
So it's above that threshold.
ranging from micrograms. Yes, yes. I'd assume. Obviously, I don't know. I don't have data from the lake,
Yeah.
but I'd presume, along with most other mesotrophic systems, that it'd be similar.
This is a real current present
Yes.
problem.
Yes. So it's actually become a lot more of an issue across the past couple of years because as mental health issues have grown in our country and across the world even, more and more people are on SSRIs and using medication that has fluoxetine in it. So more and more fluoxetine is getting into these systems. That being said, however, each species, so if we look here, they found, before I get into what I was about to say, they found that, so phytoplankton biomass, so they found some non-monatomic dose response relationships with both decomposition and phytoplankton biomass and zooplankton abundance across the different concentrations. So they find that like at low concentrations, there tends to be a decrease in both zooplankton abundance and phytoplankton biomass. However, as concentrations, so it decreases generally speaking, but then as concentrations start to get high around the 50 to 100 micrograms per liter range, then you actually see it start to increase. So around this range, they also found that like locomotion increased. So a lot of their activities started to increase. So at low concentrations, it had negative effects, but at high concentrations, they start to find almost what looks like positive effects, which is interesting. And there needs to be further studies regarding that because we're not exactly sure why that's happening. One of the guesses around it though is its relationship with temperature. So in one study, they found specifically that higher temperatures actually decrease the inhibition rate of fluoxetine. So in other words, at higher temperatures, fluoxetine has less of an effect on these photosynthetic microorganisms. So that kind of puts a stipend on how much fluoxetine really impacts these species.
Kind of a positive effect on
Yeah.
global warming.
Yeah. But on the downside, they found actually, which I thought was really interesting, that under higher concentrations of fluoxetine, the thermoplasticity of these species decreased. So they'd be more susceptible to changes in temperature. So as fluoxetine concentrations increased, these species became more, what's the word I just used? Sorry.
Inhibited by growth.
Yes, yes. But
I'm gonna have to move on here, but.
okay.
I just wanted to say how impressed I was with your study of, current real problems.
Thank you.
Difficulty here.
Oh, that's okay. So
You're hired.
we took a look at the presence of large crustaceous we took a look at the presence of large crustaceous zooplankton in Conesus Lake and Hemlock Lake. So basically we started by looking at Conesus Lake, and in Conesus Lake, alewife have been introduced to Hemlock and Conesus Lake. They've been introduced to Conesus, to the Great Lakes watershed. So what they found in a previous study that in Conesus Lake, with the introduction to alewife, the populations of large crustaceous zooplankton were essentially wiped out. And another previous study found that some large crustaceous zooplankton could still be found near shore in Conesus Lake, in some of the macrophyte beds, which are just the plant beds near shore. So we took a look to see if this was also happening in Hemlock Lake. So we looked at a species of large crustaceous zooplankton, specifically the Schizodioptimus.
Okay, so what we did is, in Hemlock Lake specifically, we compared nearshore and offshore samples. For the nearshore sample, we did a horizontal collection with the zooplankton net. And with the offshore, we did a vertical collection with the zooplankton net. Took those, preserved those crustacean, and compared sizes for each nearshore and offshore.
So what did we find? We found that on the nearshore, these Schizodioptimus copepods, they are about 6 times more abundant on the nearshore than they are on the offshore. And you can see in figure 2, the pink bar, they have a longer average length on the nearshore as well. Figure 3 shows that as well, just that the nearshore lengths are more skewed towards the right, so they have longer lengths. And we think that this longer length in the nearshore is due to there being a higher proportion of adults there. You can see in figure 4, there's a higher proportion of male and female adults, which are going to be longer. And there's more copepodites, which are the juveniles in the offshore, which are shorter.
We think there may be 2 explanations for why we see the Schizodioptimus adults in the nearshore, one of which could be a migration pattern that we haven't read anything about in the literature, that we haven't found anything yet. And the second possibility is that the adults have been preyed upon very heavily in the offshore, and then there's just more in the nearshore because the alewife are not there as much preying upon them. And then it's important to note that Conesus has a decimated population of Schizodioptimus. There are none in there. And this is because there's a high concentration or high abundance of alewife. And then if you look at Hemlock, it has about half as many for this particular outing where they recorded the abundance of alewife in catch per units. And
All right. Thank you very much. I apologize to address our Zoom audience. And
I'm sorry, I didn't hear you.
I'm going to ask you to address our Zoom audience. Okay. Give a little introduction to your poster and your project, and we'll see how it goes.
Do I have to click any button or anything, or it's
Just you're all set. Just hold it up.
like right here?
Thanks, Steve.
All right. Hi, my name is Joey.
My name is Rich.
And today we are presenting our poster on lake sturgeon and specifically the status and efforts of restoration across the Great Lakes. And so this is a lake sturgeon right here, if you've never seen one before. And they are native to the Great Lakes region. And they're a very important fossil species, being around for 200 to 135 million years. But in recent times, they've seen a decline in population and are now present on the endangered species list. So our poster is basically reviewing the reasons why they are decreasing in population and to see the methods that are being put in to restore them. And so to start off, some of the reasons for decline in population, one of the major ones has to do with the lake sturgeon and its growth rate, because they have a very slow growth rate and reach maturity at a very very longer age. So as you can see here, here's the lake sturgeon life cycle. And adult lake sturgeons, males usually reach maturity around 15 years of age, while females are even longer, reaching maturity around 20 to 25 years of age. So it makes it very hard for the population to actually recover and stabilize if they take that long to, if they have to wait that long to actually get to that age to spawn, as well as protracted spawning periods limit the number of offsprings, tagging along with the slow growth rate to make it very hard. Another reason is that lake sturgeons require a very highly specified environment to spawn in. So certain type of sediment they need, certain kind of discharge, certain temperature, all that makes it, they need very specific conditions. And if they don't have that, then makes it hard for them to spawn. Over harvesting of lake sturgeons, another big reason, as in prior years, lake sturgeon were seen as a nuisance fish. So they would get stuck in the gill nets of fishermen boats and basically break their gill net setup. And fishermen weren't too happy about that. So they would usually kill them when they caught them. And then other things such as they would use the female caviar to eat, and there would be sturgeon soup as well that they would eat. And then one of the really big ones is just habitat loss, degradation, and fragmentation has significantly hurt the population, as if they don't have anywhere to spawn in, then they're not going to be able to repopulate, as well as if there's things that are blocking their migration route, then they're not going to be able to get to the habitat. Even if there is one, they're not going to be able to get to it and spawn.
So in terms of restoration efforts, New York State specifically has put extensive amount of funding into the hatchery programs producing these fish. However, it does come with its issues, being as New York State specifically, they stock these fish at the early juvenile stage. So that comes with problems. So being as you have to, then you can't guarantee the survival of these fish for another 10 to 15 years because when we stock them, we don't stock them at sexual maturity. So these aren't spawning fish when we put them into these systems. And also given the nature of how these fish are raised in these hatcheries, being as they're not growing up in what would be a normal environmental conditions for them, it comes with a risk to disease, parasites, or just overall poor health of these individuals. However, there has also been the removal of multiple dams within the Great Lakes system that was previously blocking the migration patterns of these fish. This can help them then reach back to these sanctuary sites or where these populations go and spawn, as you can see in this figure right here. Also specifically in the Detroit River, there has been reef construction, human efforts to construct artificial habitat for these fish, going back to how they require a very specific set of conditions. By creating these habitats, we can try to aid and expedite the process of their population recovering. And also, in terms of catch and release efforts, New York State specifically does not allow catch and release on these fish. If for whatever reason you do happen to encounter one of these fish and catch it, you have a very limited arrow to actually release the fish, and you cannot take it out of the water whatsoever. We're very specific about how they're handled, just because of just how fragile the species is overall. But overall, though, despite these fish remaining endangered within the Great Lakes system, we are still putting in significant effort to bring these fish back. And I think the main takeaway here is that collaboration with government and state agencies is essential for advancing public knowledge on these fish, raising awareness for them, and just overall protecting and restoring what's left of their habitats and when they can actually spawn there.
You know, I just heard recently about a project where they were building wind turbines offshore. And what they did was they made the foundation of the offshore wind turbine mass be a environmentally friendly environment for local fauna and fish and so forth. I wonder if this would be a situation where, I don't know if they're putting any wind turbines in the Great Lakes,
I don't believe there's any wind turbines in the Great Lakes, but going back to the dams, that poses like a significant threat for the movement of these fish. Given there are fish ladders, what they call fish ladders, where it does give these fish an opportunity to get through the dam. However, that poses its own threats in its own because the way these fish ladders work is they create almost a certain back pressure when while these fish are moving through, they're not necessarily breathing if at all while going through. So if you have a larger individual with a higher aerobic respiration rate, you're not guaranteeing the survival of that fish going through the ladder. And that's also accounting for, you know, a slim chance that that fish is going to find the fish ladder before it finds the, you know, the turbine that's under the water can't be power. So
I'm talking about me, sorry.
Am I good to go?
Yeah, you're good to go.
All right, so my study did a comparison of dissolved oxygen dynamics and trophic state within restored and referenced coastal wetland habitats along the southern shore of Lake Ontario. This was primarily done in the Braddock Bay area, but also consisted of Cranberry Pond and Buck Pond. For those of you who don't know, healthy Great Lakes coastal wetlands are essential in providing habitat and refuge for both native fishes and zooplankton in addition to invertebrates. But because there's been a lot of cultural eutrophication as well as altered hydrology due to this degradation, in addition to the invasion of cattail species, also known as typha. Typha alter biogeochemical processes by forming dense mats that block access to a lot of these habitat for young deer fishes, and they deplete all of the oxygen from that. Although there has been some work in recent years by the U.S. Army Corps of Engineers to excavate these channels and create more potholes for more habitat. And I wanted to perform a comparison in nutrients on whether or not there was any merit into the restoration work being done by the U.S. Army Corps of Engineers. At each site, more notably in reference and restored channels, reference being the pre-existing wetlands, we split up some samples, around 4 to 5 per site. And we collected some data on temperature, dissolved oxygen, phosphorus. But to get into the nitty gritty of it, as you may see by some of the graphs, which may look different on your end, but the general consensus is the same, that in restored channels, there was a lot more oxygen available for these species. And this trend continued in both the months of June and July.
When you say restored channels, these are channels that have been re-dredged, or how were they restored?
Oh, they were just like the excavated,
So they dredged it up.
the dense mats of cattails that were taking over that space.
And that was inhibiting water flow.
Yeah, it was inhibiting like either growth and just blocking habitat and refuge and just taking all the oxygen that's available for these species.
Thank you, sorry.
No, you're good. I didn't know if there was a question or. In general, I'd say, again, that these oxygen concentrations were higher in the restored channels, but in June, it was more difficult to tell because it was primarily anoxic and that there was no oxygen at all in a lot of these places. But I think the trend still continues in that there's still slightly more available in the restored due to that seasonality shift. In terms of phosphorus, there was a lower mean total phosphorus in chlorophyll A in restored channels at Braddock relative to reference at Braddock, as well as for the other 2 wetlands, just in the month of June alone. We also observed the higher mean total phosphorus and chlorophyll A and restored in the month of July. But as a conclusion, there seems to be a lot of merit in these restored channels, and more maintenance should be done in order to continue this restoration work.
Very good. Thank you very much. Appreciate it.
So this semester for our research project, we've been looking at amoeba herbivory in Conesus Lake. Blooms of these small single-celled photosynthetic bacteria have been observed since 2010 in the lake around July. and we've been curious about what kind of organisms have been eating them. We found that the lake is absolutely teeming with these amoebas called diflugia. Down here, figures 1, 1.2, and 1.3 show an illustration and a couple of pictures of diflugia. We tested the hypothesis that the diflugia are ingesting these tiny cyanobacteria, which are in the same group as the cyanobacteria which cause harmful algal blooms, also known as blue-green algae. The number of these tiny cyanobacteria and microalgae were compared to their abundance in the lake, and we hypothesized that the amoebas could be used as a model to predict the phytoplankton abundance in the lake.
So we collected our data using a very fine mesh net that we towed behind a boat. And then we analyzed the data underneath a microscope. First a regular microscope and then an electron microscope. And we were able to differentiate between the sizes of the cells within it. So small were 1 to 2 microns, and and then large were measuring 4 to 6 microns, and we classified the orange fluorescence as cyanobacteria pigments, and then the red fluorescence was the chlorophyll from microalgae. And these organisms actually eat using the appendages called lopopods that they actually stick out and grab their food. And this can be seen over here. This is them actually grabbing the food and ingesting it. And they have a hard outer shell, and this is a bottom view, and this is the mouth where those appendages come out.
I don't know if I need to step over here. Okay. So basically, when we were analyzing the data, we found that in that figure 5 that you guys can see, that they actually correlated really well. So the orange line would be the amount of that picocyanobacteria, that small cyanobacteria in the lake. And then the blue line is how much average that we found inside of the amoebas that they were eating. So you see that there's a peak on July 13th, and you see there's a peak around July 24th. And we ran a correlation test basically just to see if it was statistically significant, which it was. And it really just is consistent with the idea that the amoebas, Lopopodia, reaching out, passively grabbing the food around them, just correlates with how much they're eating.
So we counted the number of phytoplankton in each amoeba. And while doing that, we noticed that in figure 7 on July 13th, there was a higher peak of number of phytoplankton in the amoebas, which matches with what we got before, with one of the peaks being on July 13th. And our blue measures are smaller phytoplankton, while the red measures our larger phytoplankton. So as you see in figure 7, there's a peak on the 13th. However, when looking at if this has nutrition and substance for the amoebas, these smaller phytoplankton actually don't. We calculated based on the biomass that the amoeba needs to eat about 40 small phytoplankton to be sustained by also eating 1 large phytoplankton. That's why on 713 there's a lot of these smaller phytoplankton, but it wasn't as sustainable for biomass. That's why in figure 8, the second column's a lot smaller compared to the other columns where in August where there's more larger phytoplankton, they had a larger biomass.
Are there other microorganisms that eat the cyanobacteria?
We assume so. These are the ones that we decided to study because there isn't much known about them. So Dr. Bosch actually kind of encouraged us to be looking at these guys because there wasn't much known about them. But we assume that this would be a good model for seeing other.
For next steps, we'd be interested to look at what other organisms in the lake are feeding on these picoplankton and picocyanobacteria. We saw definitely dinobrion and rotifers that also eat these, but we didn't study those because the amoebas were much more abundant in our samples.
Good, thank you. Nicely done. It's just so nice to see studies on our little lake here. They go into such great detail. Fantastic. Thank you.
So my name is Brayden Link. This is the results of my first year of my master's thesis. So I focused on Braddock Bay. So Braddock Bay is a coastal wetland about 10 miles outside of Rochester. So about 10 years ago, Monroe County and the Army Corps of Engineers collaborated to actually restore this area. Because what happened is that Braddock Bay naturally had a beach that helped protect the area from wave action erosion from Lake Ontario. However, that beach was eroded away eventually, and we lost over 100 acres of wetland in the area. So the Army Corps of Engineers came in, they restored a lot of the plant matter. They also built a man-made barrier to protect the bay from wave action erosion from Lake Ontario. So the limnology department at Brockport, we've been sampling the area before, during, and after the bay construction, or the barrier construction. So after the barrier was constructed, what we were seeing, we actually saw an increase in water quality. However, over the past 3 or 4 years, we've actually seen a severe decrease in water quality. And we've actually seen a lot more harmful algal blooms that occur in the bay. And we don't know why we're seeing them. So my thesis is looking at trying to figure out why all of a sudden we're seeing the harmful algal blooms. So what I've done is I've actually started sampling all 8 of the original sites that were sampled. I took the water from each site. I added different amounts of nitrogen and phosphorus. So I had about 10 treatments that all had varying amounts of nitrogen and phosphorus. I let the flask sit for 4 days, and then I filtered them for chlorophyll. Basically seeing like if I had a lot more growth, a lot more algae growth from flasks that were fertilized with nitrogen, so that means the area is nitrogen limited. Same thing with phosphorus, but if I saw a lot more phosphorus that caused algae growth, and we knew the area was limited in phosphorus. Basically what this tells us is that it gives us an idea of what nutrients are coming into the bay and how we can potentially limit those nutrients. So at the end of my 2 years, hopefully I'll have a product to help Monroe County actually manage the area to the point where we're not seeing these harmful algal blooms anymore. So yeah.
Fantastic.
Thank
You
you.
have a project that will actually
Hopefully have an impact.
do something, right?
Yeah.
Improve the situation. In Conesus Lake, we talk about internal loading and external loading. I understand the internal loading process is for phosphorus in Conesus. What's the process is our process for internal loading of nitrogen?
So nitrogen, I'm not as familiar about the internal loading process for that as I am with phosphorus. However, I do know there is some nitrogen that is also stored in sediments, kind of similar to phosphorus. So I'm not entirely too familiar with internal loading of nitrogen in Braddock Bay. I know we've talked about potentially adding on sediment sampling for next year to see what sort of nutrients are actually stored in the sediments in Braddock Bay. I do know that there's a lot of nutrients that come from the 2 tributaries. So next summer, I'll actually also start sampling the 2 tributaries as well to see like what's coming in, because Monroe County never actually sampled the 2 tributaries before completing the restoration project. So we actually don't know what's coming into the bay right now.
Well, thank you. I really appreciate the studies where.
All right. Hello, everybody. My name is Macy. I'm a grad student at SUNY Brockport, and I'm doing my thesis on Conesus Lake. Specifically, I'm looking at nutrient limitation within the lake across the 3 different basins. So Conesus Lake has a north basin, which is 10 to 14 meters deep, a center basin, which is about 12 meters deep on average, and then a south basin, which can reach depths of 18 meters. And the North Basin is polymictic, which means that it mixes multiple times throughout the year. And then the center and south are dimictic, meaning it mixes once in the fall and once in the spring. And these mixing events bring nutrients from the sediment of the lake up into the water column. And the nutrients are also being added from the tributaries as well. And the nutrients I'm interested in specifically are nitrogen and phosphorus.
So I set up an experiment with the goal to determine how phytoplankton, which is algae, is limited across Conesus Lake in the summer months, July and August. So for my methods, I did a laboratory experiment with 9 treatments. You can see that in the table at the bottom. Treatments 1, 2, and 3 have low P. Treatments 4, 5, 6 have medium P. And then treatments 7, 8, 9 have high phosphorus. And then treatments 1, 4, and 7 have an N to P ratio of 2. Treatments 2, 5, and 8 have an N to P ratio of 20. And then treatments 3, 6, and 9 have a 40 N to P ratio. And I use those treatments in flasks. So 10 treatments for each basin, and I replicated that 4 times. And I took water from each basin in July and August, and I put them in an incubation chamber with 60% humidity, 24 degrees Celsius, and 16 to 8 light dark cycle. And I stirred them once a day for 4 days. And at the end of that 4 days, I took chlorophyll analysis. And that is what these graphs are showing. That is showing the average and based on the treatment. So the letters represent the basin, and then the numbers represent the treatments that correspond to the table. So for July, you can see that treatments 8 and 9 across all basins had the highest growth of phytoplankton or algae, which had 20 or 40 N to P ratios and had high amounts of phosphorus. And then in August, I saw in the center basin treatments 5 and 6, which had medium P, and again, high N to P ratio of 20 and 40, had the biggest growth. For August, the North Basin saw treatment 8, had the highest amount of growth. And then in the South Basin, it was treatment 6. So one reason for these differences, I suspect, is that there are different communities of algae growing by the months in Conesus. So I actually took samples from my flasks and I preserved them. And I'm going to look at them under a microscope to hopefully identify the different algal communities and see if that explains the variation in nutrients. And I'm going to run this experiment again in next year.
Oh, when you talk about treatments, these are, can you say a few words about, I mean, what that implies to me is something we can do to fix it.
So
It's not really what you're talking about.
No, the treatments are just the different amounts of NP that I added. So I call them treatments to easily distinguish between each one.
I wanted to see, it's not like
No.
medicine for the
No,
leg.
it's not medicine, no. If you wanted treatments, my treatments that were the low in N to P, which is that 2 to 1 N to P ratio, are ideal for keeping, at least in July and August, for keeping the algal mass low.
Thank you very much. I'd love to see these projects on Conesus Lake. Great detailed information with solid science behind it. Well done. All right. Well, last but not least. Absolutely. I got the right slide. Everything's working well.
All right, we good?
You're good. So
all right,
introduce yourself. And
I'm Jackson, and we got Nick and Chris here, and we studied the question: are nearshore harmful algal blooms in Conesus Lake caused by horizontal wind transfer or by local conditions? So we wanted to expand on that and ask: do the harmful algal blooms originate near the shore, or are they blown to the shore from offshore by certain wind conditions? So our hypothesis was that the species composition of nearshore HABs, harmful algal blooms, should be very similar to that of lake-wide communities, because harmful algal blooms are caused by wind-driven conditions that concentrate cyanobacteria from all across the lake. And I'm going to hand the talking stick over to Chris, who's going to take you through the methods and results. Sure.
So first off, we collected samples from a number of different sites around the lake on 2 different dates. We looked at the species of cyanobacteria that we could identify in each sample, and we calculated the relative proportions of species at different sites around the lake, and calculated what's called a percent similarity index between the communities at different sites. And if these are lake-wide conditions, then the communities at different sites around the lake on the same date should be very similar. We found, in fact, that they were very similar. Our lowest similarity was 68%, and most of them were in the 80s, up to 90% similar between different sites. Most striking was that on the 12th, there was a sparse cyanobacteria sample from the offshore down at the south end of the lake, and there was a much more concentrated bloom at the north end of the lake. And when we looked at the cyanobacteria communities between those 2 sites, they were 82% similar, despite the distance between them, and despite that one was shoreline and the other one was out over the deep water. So how that ties into our hypothesis, I will hand over to Nick.
Hello, I'm Nick, and I looked at the raw wind data collected by Conesus Lake. And for the 12th and the 19th, which were the collection days that we did, I got wind direction and wind speed for both of those days. And what I found, which is what these graphs are based on, is that on both days, before collection time and during collection time, the wind direction originated from the southeast, and the wind speed was calm in the morning, which allowed cyanobacteria to rise, and the moderate winds in the midday around the collection time pushed the cyanobacteria northward from the southern originating winds. And this is what explains the fact that the southern location was very sparse, but it also shared a similar composition to the northern location, which had very dense. So what we concluded is that the southern originating winds pushed the cyanobacteria from the south northward, creating the blooms in the north.
So if prevailing winds are pushing in cyanobacteria from the center of the, wherever it's coming from, would we be able to use wind information to predict? I mean, is that the idea?
Yeah, I think that's a very big part of why this is important, because I do believe we could use the wind data that is collected at Conesus to predict where the blooms will appear. So if there's a wind originating from the north, we may expect the blooms to be pushed south. And if there's southern originating winds, like we saw in our data, there's going to be blooms showing up in the north. So I feel like it's a good predictor.
So, my understanding is that cyanobacteria, some species have the ability to kind of control their buoyancy and therefore their depth. They would need to be near the surface in order for this to be a factor. What's your thinking in terms of how many, what kind of HABs have this ability to control their depth, and those that do, or maybe those that don't, how do they get modeled?
Yeah, so the buoyancy is absolutely a factor. The 2 species that we saw, the vast majority at our sites were these 2, Dolichospermum lemmermanni and Dolichospermum circinale. This one, the lemmermanni, was by far the most abundant. And one of the things about this is that it forms these giant clumps of filaments. And the way they form, because they're all tangled together, they have the ability to trap gases inside of them and to trap oxygen inside of them. And so that really helps them float to the surface. And so that's really just a subset of cyanobacteria that have that kind of buoyancy property to them. So really, although we see a few other species in with our samples in small numbers, really the ones that we should focus in on are the, our thinking is the ones that have that buoyancy, that can come to the surface and then be driven by winds and accumulate along the shore.
So there's this, some we'll be able to predict using this model and some we won't.
Certainly, that's the hope, yes.
Very good.
Thank you.
Well, I love seeing projects on Conesus Lake, especially the projects that can go to such detail. And really, maybe then you can give us some real tools to deal with some of these.
Yeah,
Thank you. I think that's one of the major reasons that we were interested in this project particularly is because it's a very local problem and because it's a problem that's of such interest to everyone, in the area. It's a real concern. And so looking at how to,
The local problem with global implications.
Absolutely. Yes.
Yes, and we
So-
hope that studies like this will help with prediction and management.
Yeah, cool.
So
Thanks, guys.
Absolutely. Thank you.
Well done.
Thank you.
All right, I guess that's the end of our Zoom presentations for the various posters. Nice to see all this great work done by these young people at both SUNY Geneseo and SUNY Brockport. Particularly nice to see projects that are helping us understand Conesus Lake, since we're so tied and close to the area. So thank you for joining me. I think I'm going to be getting off of Zoom here, and we'll talk to you next time.
Thank you so much for coming. Great work. Those posters were great. I learned a ton. Again, I'm the watershed inspector for Conesus Lake. If you ever have any questions or need anything, please give me a call or email me. We have some certificates for you as a thank you. Again, we really appreciate you coming. Thank you, Carl, for all your hard work. I think the folks on Zoom had a pretty good experience. It sounded really great. I'll turn it over to Mary to talk a little bit about our programming here at the WEC and can be done.
Yeah. Hi, I'm Mary Underhill, and I'm the Conesus Lake Watershed Manager, and I do work with Don closely. I do greatly appreciate the work that everybody has done, the CLA, to support the students in SUNY Geneseo and SUNY Brockport. We really love the work that you do, and we look forward to this program. We hope to see some of you guys in the future. We do have several programs coming up. We've got a whole lineup for 2025 built out already. So we're inviting folks to come to our January program. RIT and U of R students are presenting on microplastics, and that's a nice support for your project. And in February, we have deep sea diversity and adaptation. I won't go any farther than that, but it is listed on the CLA website, and we're hoping that people come to the Watershed Education Center either in person, we're in Vitale Park, or join us virtually. Well done, everybody.